This is part 4 of 6 for the immune system. This part is going to focus specifically on humoral immunity. So before we begin, let's take a look back at our outline here. And so once again, here we'll focus on humoral immunity. And specifically, when you think about humoral immunity, what I want you to think about again are B lymphocytes. So when we go through humoral immunity, the big picture here is that we're going to be able to take B lymphocytes and we're going to be able to produce plasma cells, and those plasma cells are going to be able to produce antibodies. And then we'll be able to discuss what those antibodies are going to do. So you may want to add to your flow chart here the function for those antibodies, and there's several things that the antibodies can do. Now, B lymphocytes can also get converted over to memory cells. Memory cells then, when they get exposed to a particular antigen once again, like a second time, then those will go on to produce plasma cells, which then produce antibodies, which then can help mount that immune response. Okay, so that's the overall picture of where we're going with this. So again, just to kind of give you the same idea again, when we talk about these B cells. So let's take a look at these B cells that are shown here in this top part of the diagram. These are B cells that are specific to certain antigens. So if you notice the one here that has the red antibodies, it's very specific to these red antigens, right? So when the red antigens bind, and do you remember what they're binding to? What's the name of this receptor? This is called a membrane antibody receptor, or we can call it a membrane immunoglobulin receptor. Okay, so when this happens, then what your B cells are going to do is they're going to proliferate. So they can become plasma cells and memory cells. All right, so off to the side, let's kind of diagram this big picture as we go. So here we have our B lymphocytes, and they have to bind to an antigen. And remember that they're binding to the antigen by using the membrane antibody receptor. And once they bind to that antigen, then they can become either plasma cells or they can become memory cells. Now, if they become plasma cells will actually give rise to antibodies. So literally they produce antibodies. Memory cells, when you're exposed for the second time, so you have a second exposure to a particular antigen, those will go on to make plasma cells, which will then make antibodies. So it's much faster when you're exposed to an antigen for the second time because you have these memory cells that are hanging around waiting for that antigen to be, waiting for your body to be exposed to that antigen once again. Now, plasma cells are short-lived. These do not live for very long, so these are short-lived. Memory cells are longer-lived. They're long-lived. Okay, so they survive for a much longer period of time. So this is what we call the clonal selection theory. So what that means is you have these B lymphocytes that give rise to a whole bunch of plasma cells that are all the same, and those plasma cells all produce the same antibodies. And the B lymphocytes also give rise to memory cells, a whole bunch of them, so they're clones, right? And then those can ultimately give rise to more plasma cells, which can give rise to antibodies. All right, now, even though we're discussing these with respect to B lymphocytes, keep in mind that T cells can also do this, but it's not something that we're emphasizing in this course. Okay, so plasma cells then, we know, can produce antibodies, as I just talked to you about, and they will combine then, those antibodies then can combine with a specific kind of antigen. So I'm going to show you here in just a few minutes how that will work. Now, when the antibodies enter the blood, they're known as GABA globulins or immunoglobulins. That's where that term immunoglobulins comes from. So we have several different subclasses of immunoglobulins, and here you're just going to have to memorize these because some are found in different places. So we'll use some of these as we go throughout the next series of slides. All right, so how does the antibody actually destroy the antigen? Remember that our bodies produce antibodies because they want to destroy the antigen. So here are the methods that they're going to use: neutralization, agglutination, enhancement of phagocytosis, activate the complement system, and they can stimulate killer cells. So be sure you've watched the previous videos so that you understand phagocytosis and complement and natural killer cells before you continue on here. Otherwise, you won't quite understand what's going on. All right, so we're going to cover each one of these. So remember the big picture. We want the antibody to destroy the antigen. So ways to do that. Let's start off with neutralization. So keep in mind that as we go throughout these slides, when you see the term pathogen, the pathogen is the antigen, right? That's the foreign invader. So in neutralization, here you have your antibody, okay? And that antibody binds to the pathogen. Notice how many antibodies are bound to this pathogen. There's a whole bunch. So because you have all of these antibodies bound to the pathogen, that's going to block the activity of the pathogen. And if you block the activity of the pathogen, what you've essentially done is you've neutralized it. The antigen is unable to invade the tissues. All right, so in this case, that antigen cannot invade that target tissue because it's been neutralized. It's got all these antibodies that are bound to it. Now, this isn't going to necessarily kill the pathogen. It could, because some pathogens, if they can't get into a target tissue, can't get the nutrients that they need to survive, so it might kill them in that way, but it wouldn't directly kill them. Later we'll talk about how we actually kill the antigen. Now there's also agglutination, and this is something that you should already be somewhat familiar with when it comes to blood. When we talked about blood antibodies and blood antigens. So in this case, we're going to be dealing with two different types of antibodies. We're going to deal with IgM and IgA. So these are two different immunoglobulins, and what these immunoglobulins do is literally they clump the antigens together. All right, so here you see all of these pathogens, and it's all clumped together, and we're using antigens to clump them together. Because if you clump all these antigens together, then that blocks the activity of your pathogen. So that pathogen, for example, couldn't invade a tissue. All right? Be sure you understand agglutination when it comes to blood types as well. Okay, then there's opsonization. So this is where we start off. Here's your macrophage, which is a type of phagocyte. So opsonization can occur with different types of phagocytes. Here we're focusing on a macrophage. So you have an antibody that binds to the phagocyte. And when the antibody binds to the phagocyte, then the antibody binds the antigen. And what that's going to do then, it's going to trigger opsonization. So in other words, this is triggering phagocytosis of the antigen. All right, and if you recall from earlier, opsonization enhances the speed, or it enhances phagocytosis, in other words. So it's a lot quicker. Makes sense if you want to phagocytose a pathogen because you want to start killing off those pathogens. Now there's also complement activation, which we talked about earlier, but this is a different way to activate the complement system. So we talked earlier about having a bacterial cell that was our pathogen, and how there was a polysaccharide component on the cell membrane, and that activated the complement. Here, however, we're using a different method. We're going to have an antibody that's bound to the pathogen. So here you can see the Y-shaped antibody, right? And it's bound to the pathogen. So then the very first complement protein recognizes that. And when it recognizes it, then it binds to the antibody. And then that triggers the complement system. And the end result of the complement activation system is that you develop this pore, which, if you remember, is called a membrane attack complex. So if you develop a membrane attack complex, in other words, a pore in the membrane, how does that kill the pathogen? Remember that once you have a pore, that allows for increased permeability. So you have flooding in of ions and water into the cell, and the cell is going to lyse. All right, antibodies can also enhance natural killer cells. And so if we look here, here's our natural killer cell up on the top left. Our abnormal body cell, maybe it's a tumor cell, is on the bottom right. So what we're going to have here, we're going to take an antibody and it's going to bind to the abnormal cell. So basically when it binds to this abnormal cell, it's marking the cell for death. So when it's marking it for death, what that means then is that the natural killer cell will release a chemical that you're very familiar with, and those are called perforins. And when the natural killer cell releases perforins, that creates a pore or a hole in the membrane of the abnormal cell. So we know from earlier that if you create a hole in the membrane, you have increased permeability. In comes the ions, in comes water, in comes all sorts of goodies, and the abnormal cell will lyse.